Good. Woohoo.
Oh, fame at last.
Champapo.
Right, let's share a screen then.
Do you have a camera? Or?
Yes. Is it actually on at the moment?
Okay.
Yeah, there should be a camera.
Okay, we don't see you.
It's just.
But it's okay. Okay, so everyone now, everybody's arrived to this Zoom. So I can see that there's a room for me improving my the way I formulate these questions in the quiz that we had. But anyway, you're slowly, I think, getting it. So we're on the way to something very good. We will continue later. The Zoom, the quiz, I want you to finish it. So if you have any questions, please put them in the discussion forum so that we can sort this out. And now we're going to talk about colocalization. So it's another off the way topic that Jeremy Adler, who works at BioVis in Uppsala and has published several papers about colocalization. He's going to tell us about it, and then we'll have a little demo or a little workshop afterwards. Please, Jeremy.
Right, let's just get the screen under control.
It's okay if we don't see your video, but maybe if you can share.
Yes, I'm about to. Right, are you seeing a screen?
No, we don't see your screen.
Okay, it's not good. Right.
But you can share your desktop in general. If you share the desktop, it does happen somehow. Something is happening. All right, so I think you're
good.
Maybe seeing your desktop, but yes. Good.
Right, I think, are we in business?
Good. Yes, thank you.
Right, so the opening screen is obviously not a confocal image, but it's representative of science in general. We fight, but we fight by throwing books and documents at each other. As far as colocalization goes, what we're going to try to do is work on some definitions, then consider how we're measuring it, and then follow that up with some kind of hands-on session, or at least a demonstration of how we might mess around with pictures. So the first issue, obviously, is image acquisition. I've assumed basically that this has been covered before, so there are lots of very standard things. The images must be aligned. An important one is each image should only be formed by a single fluorophore. Otherwise, we get wonderful colocalization for completely the wrong reasons. Image quality will get mentioned a number of times. This has two issues: resolution and signal to noise. Obviously, we need all our intensities within the response range. And another one that doesn't get mentioned often is even illumination, especially in a wide field system. If parts of your area being imaged are more intense than others, then you will get intensity variations that have nothing to do with colocalization. They're due to uneven illumination. Right, so we have some pictures. And so what we're seeing in the first one, two grayscale images, they're obviously very similar, but they're obviously not identical. So what we're looking for is some way of quantifying or deciding how related these pictures are. We can see that image 2 just seems to have a plasma membrane, whereas image 1 probably has a plasma membrane and a nuclear membrane. In this second panel, we're showing two images in false color, where parts of the plasma membrane are present in one image and most of it is present in the other. So the issue is, what measurement might we make, and how can we make sure that our measurements are accurate? I mean, a worrying thing with images always is, or measurements of any sort, is that you have a nice group of decimal places, but what doesn't get considered is, are the numbers actually right? In other words, do we have nonsense plus decimal places, which is more than possible. So the kind of questions, overall, we're looking at distribution and relative distribution. So we might, at its simplest, measure the area occupied by each fluorophore alone, and then the area that they have in common, simply as a fraction of the total image or something related to that. We might move slightly more upscale by saying we could look at the amount of each fluorophore that is found in the presence of the other. That's moving from simply counting pixels to looking at the total amount of fluorophore. We might ask more complicated questions. If they're not in the same place, are they actually near to each other? Again, these are all different questions. So when you're starting out, when you're capturing your pictures, you want to decide what measurements you want to make, and therefore to get images that contain sufficient resolution to support the measurements. Again, we might be looking at distributions within a tissue or within a cell, or distributions relative to a structure. Are they congregated around the nucleus? Are they perhaps more distributed in the location of mitochondria? At the ultimate, you might say in a mitochondria, are we finding it inside the mitochondria or in the mitochondrial membrane? So these are all distributions. We might then further complicate matters by considering, is there an intensity relationship? Where one fluorophore is abundant, is the other one abundant? Or is there no relationship, or even possibly a negative relationship, where one is abundant, the other is absent? But to start off with, the classic approach to looking at colocalization is simply to superimpose or to overlay two images. The classic red plus green makes yellow. So here we're showing two pictures on that basis. Both contain a red and a green image, and when you immediately look at them, they appear to be different. But they're actually the same pictures, except that we've messed around a little bit with the brightness and contrast. What this is saying is that you can display things in an informative way, but there is considerable scope for choosing and therefore bending what is presented. But of course, this is only a picture. It's not a measurement. Measurements are harder to distort than simple pictures. If we consider overlays again for a moment, here we're showing a full range of red intensities, here a full range of green intensities, and then when we combine them as an overlay, we have every possible red and green combination. So if we were to define colocalization as being yellow, where exactly is yellow in this? Or at least, where do the limits of yellow end? This is always a problem. It's further affected by the monitor you're using. It might also be affected by your own eyesight. Do you register red and green equally? There are lots of problems with display, but display is better than not displaying it. But what we really want are measurements. Alternatively, in displaying, there is some push now to use blue, maybe blue and yellow, to help out the small group of men who predominantly, or at least who are about 5 to 6% who have problems with red-green discrimination. So maybe blue and yellow work for you. A problem with all of these overlay methods is that down here, where the intensities are similar for each fluorophore, the color disappears depending on how your screen is set up. So you can lose or fail to see information. And of course, it's generally picking up this 45-degree angle. Yeah, which picture's yellow, which pictures are white is very subjective. And of course, it's very easily photoshopped, as we've demonstrated, and we'll demonstrate again here. Red, green overlaid. If I mess around with the offsets, I can hide the fact that there is considerable presence of each fluorophore within the cell. So definitions. The area fraction, the number of or fraction of pixels where both fluorophores might be found. The M1 and M2 coefficients, which are probably the most popular. Here, what they're looking at is the fraction of each fluorophore found in the presence of the other in some biologically meaningful region of interest. They're also the K1 and K2 coefficients. I've no idea what they measure. I don't think anyone has ever clearly explained what they measure and why it might be considered useful. There is the Manders overlap coefficient, which is a kind of cut down version of the Pearson correlation coefficient. And there are many more. And even more depressingly, people keep inventing new ones. A really important consideration, apart from which coefficients you might use, are a region of interest, which pixels to include, and another question, should you include pixels that only have background intensity? Again, all of these things will affect the results you get. It's a kind of world of coefficient roulette when measurements are not clearly defined and are not clearly explained in a method section. For instance, Manders refers to at least 3 different coefficients. So if you say, I use the Manders co-occurrence coefficient, what do you mean? Even at this very minimal level, papers are often unclear. So a number of years ago, looking at this massive colocalization coefficients, we proposed the idea that we could split colocalization into 2 major measurements. Co-occurrence, which covers their appearance or presence in the same place, and correlation, which discovers or which considers the relationship between intensities. The major point is these are totally different measurements. Ideally, you should measure both. Which one you would choose or whether you would make other measurements really depends on your underlying scientific question. But the major point is they are totally different. Here is just a mess, really, a Venn diagram showing coefficients that work for co-occurrence, those that work for correlation, and then there's a group in the middle that measure a bit of both. These are basically hopeless. It's simply because it's impossible to clearly state what they are measuring. If we consider the world or coefficients can be defined as co-occurrence, are they in the same place? Correlation, what is their intensity relationship? So here we have 2 images, and we're measuring co-occurrence using the Manders M1 and M2 coefficients. So what this is saying is that on a scale of 0 to 1, roughly half of the pixels are present, or half of the total intensity of one fluorophore is found in the presence of the other one. Then flipping it round, almost 90% or 0.9 of this fluorophore is found in the presence of the other one, which isn't really surprising. This one is present, it looks like, in most of the plasma membrane. This one is only present in part of the area. But the critical thing is that we make the measurement from a region of interest, which begs the question about how we actually define the region of interest. If we now consider correlation, what we're primarily, or at least a primary way of looking at correlation is to create a scatter plot, something that's very common in flow cytometry. But essentially what you're doing is for each pixel within the region of interest, you're finding the intensity of one fluorophore and plotting a point that marks the intensity of that fluorophore, how it corresponds with the other one. And in this case, we see a scatter plot with no obvious orientation, which is consistent with the measurements for the Pearson correlation coefficient, which is close to 0. The scale is -1 to +1. The SRC would be the Spearman ranked coefficient, which is very similar to the Pearson, except you rank the data before making the measurement. And in both cases, the values are about 0, which is entirely consistent with what you see. But it's important to look at the data, to look at the images, and not simply believe the numbers that come out. So let's look slightly more detail about the Manders M1 and M2. This would be the equation, the sum of red intensities in pixels where the other one is present as a fraction of the total red intensity. And the same for the green. It's a nice, simple, intelligible measurement. But here's a question. What would happen if distributions were random? Say, for instance, the red fluorophore occupied 20% of the region of interest. The green fluorophore occupied 20%. We found both present in 18% of the area. How does this differ from a random distribution? In other words, we're raising the question is, are the distributions we're seeing different from random? If the distribution is random, we don't have to look for an explanation. If something is non-random, then there is an underlying cause that may be biologically interesting. So in this case, what would the random overlap be? One fluorophore occupies a fifth of the area, the other occupies a fifth. So if they were distributed randomly, we'd expect a fifth of a fifth, roughly 4%. So if the measured overlap is 18%, this is way above what we'd expect randomly. But any measurements made of area are very dependent on thresholding, how we decide which pixels do have the fluorophore present and those that don't. This is a major problem in almost everything. So what we're showing you is scatter plots, and we're showing the histogram that correspond to the scatter plot to give you some idea of where we might, if we were doing this manually, put the threshold. Here for the red fluorophore, we're putting a threshold in a nice plausible looking dip, and there is a less plausible one for the second fluorophore. What we're trying to do here with thresholding is differentiate between the pixels where our fluorophore is present and those where it is absent. And this is not intrinsically simple, but it has a huge effect on the measurements. So if we're looking for co-occurrence by area, what are we actually going to be doing? We're going to be taking our green fluorophore, thresholding it, our red fluorophore, thresholding it. Then by combining these two binary pictures, what's called an AND operation, we can define an area where both fluorophores are present, and then make, if we're simply sticking with area measurements, the area of each binary image may be sufficient. We may be able to define more interesting regions of interest, like this would appear to be the nucleus. This may be the cytoplasm. How well we can define these is obviously quite important. For correlation, we're considering, is there an intensity relationship? Where one is bright, is the other bright? So if we look at this pair, we can see where this one is present, the other one also tends to be present. So if we measure correlation, it's 0.65, which is at least on the right side of 0. In this second image, we're seeing a much lower correlation, 0.35 or 0.335. So it's saying that there is a relationship between the intensities, but it's not as strong as in this image. Whereas in the final picture, we're seeing something quite unusual, a negative correlation. Sometimes questions about what a negative correlation means are raised, and several people have made attempts to avoid ever recording one. Nominally, at least, it means avoidance. What it's saying is that where one fluorophore is present, the other is increasingly absent. But it's hard to think of a biological mechanism that will produce avoidance. Molecules are known to associate, but there are no mechanisms for short-term avoidance. So avoidance is used as a word, but it doesn't explain anything very useful. Later, we'll see why we might get real negative correlations. When we're testing coefficients, when we're looking at whether our measurements are correct, What is very useful is to generate sets of data where you know precisely what is going on, which is what's done in this case. We have some ground truth data sets. We can see this is quite a strong negative correlation. This is a strong positive one. And then in between, we can generate different strengths, with 0 being no relationship at all. This is then useful to test against any measurements you make. This is a ground truth. What is the measurement? Does it actually reinforce the truth, or does it report something different, which we'll find later happens? So if we have a reasonable positive correlation, we might consider this to be that the two fluorophore molecules have an affinity for each other, or an affinity for a common structure. The common structure could be something as simple as a membrane, but say within the membrane, there might be no association between them. If the correlation is 0, we're saying they're found together, but there is no interaction either directly or with another structure. Negative correlations, as I've just said, are slightly trickier. They're found together, but show a degree of avoidance. So here we're looking for some kind of mechanism. Let's have a look at the Pearson correlation coefficient. It's the most common correlation coefficient, and it's surprisingly, as an equation, quite neat. The important thing, it has a range. 1 is a perfect correlation, 0 is a random distribution, -1 will be a perfect, but inverse, but probably more precisely negative relationship. So let's look at the equation. Ri and Gi are the intensities in individual pixels within whatever region of interest we're working with. Rav and Gav are the average intensity within the region of interest. So if we look at the upper part of the equation, for each red pixel, we're subtracting the average red intensity and multiplying this by the green intensity of the same pixel minus the green average. The bottom part of the equation calculates what the greatest value could be if the two were perfectly correlated. So the bottom here is simply to create the range from -1 to +1. Yes, this looks nice and wonderful. This shows how the numerator is working. This is a scatter plot, so values from 0 to something. The mean would be somewhere in the middle. And this is the weighting of pixels in different locations, going from negative to positive. The denominator, the part below on the bottom half of the equation, also has a weighting, but this is weighted simply by distance from the center or distance from the mean. But when we combine the two, we end up with this. Here's our scatter plot. Things or pairs of intensities up here make positive values because each is above the mean. The mean is in the center. But if both are below the mean, we have two negative numbers. Something minus the mean multiplied by itself becomes positive. But along this diagonal, everything produces negative values. One fluorophore is above the mean, but the other one is below the mean. So this is how the equation is working. It's looking for combinations in this range with the weighting that we can see here, and then subtracting combinations in that range. If things are distributed randomly, the net result is 0. One of the important questions in measuring correlation is, which pixels should I use? The critical question is, should I include pixels where neither fluorophore is present? Should I include pixels where one fluorophore is present, but the other one is not? So to examine this, we decided to generate some sets of images where in image 1, we simply throw in some random blobs where their position and intensity is randomized. We do the same for the second picture, just two sets of random blobs. So as a ground truth, we think the correlation here should be 0. both have been generated randomly, therefore there should be no underlying correlation. Now what we're doing here is showing the correlation measurements for increasing numbers of blobs, more and more blobs added, and we're trying to find out which one is coming up with our expected value of 0. And here we can see one set only does. The AND pixels, the pixels where one fluorophore and the other one is present. They produce the ground truth result. Anything else we do, if we use the AND pixels plus we also include pixels where neither fluorophore is present, we get this weird relationship. If we use pixels where one or the other or both are present, we get this. So the only one that actually produces the ground truth known answer is to measure correlation using the pixels where both fluorophores are present. That works. The others do not. So this is a very empirical way of deciding it, but it should work. It should be true, which is useful. Some more considerations of the Pearson. What we're primarily looking at here is a scatter plot where we're showing all sets of data. The data is essentially the same, except we've changed the absolute intensity, which would be something you could achieve in imaging if one of the images is acquired for longer. You would capture more photons. So it's the same image, but effectively with different gains or longer acquisition times. So the question I'm throwing at you is, when measured, do these 4 data sets have the same correlation? I mean, you looking at them, you think, nah, they don't. This one has a much shallower angle, this one has a steeper angle. But when measured, they have the same correlation. Mostly because the calculation involves the subtraction of the mean. I mean, they won't have the same angle of regression line, but the correlations are identical. So this is important because otherwise changing the gain or changing the image acquisition time or the excitation intensity for one fluorophore would change the measured correlation. It doesn't. Same question, 3 data sets that are, in this case, identical, except one of them has been offset. Does this change the measured correlation? And again, the answer is it doesn't, because the important part of the correlation measurement is for every pixel, the value is subtracted from the mean. So if things are offset, the mean is also offset. So the measured correlation will be identical. But clearly these mean slightly different things if they're true. If they're true, and the offset is real, that there is always an intensity of 1, then the biological meaning of these 3 would be different, even though the measured correlation is the same. The important thing about the correlation is it picks up on how the variability of one is related to the variability of the other. It doesn't factor out, or it does factor out offsets, but offsets could be quite meaningful. If this data set, this one at the bottom, by extrapolation goes through 0, the other 2 don't. So let's generate some problems where things can go wildly wrong. So here we have a nice fake cell. It has a nucleus. Here we're showing the scatter plot for pixels in the nucleus, and they're basically uncorrelated. We do the same for the cytoplasm. They're uncorrelated, but have higher intensities. If I then decide to measure the whole cell, I'm combining the 2 distributions together, and suddenly I have an amazingly high correlation. Mathematically, this is completely correct, but our interpretation of this would be completely misleading. It's artifactual because we have 2 quite different regions of interest, with each having a random distribution, but when combined, we have a non-random measurement. It's important to choose your regions of interest. We can see this with a real cell, where if we look at the region of interest here, the one where we measure the whole cell, the Pearson, is higher than for any other compartment. So we would argue, or at least I would argue, that making measurements outside of or combining biologically meaningful compartments produces very, what would the right word be, misleading values. Always look at your data set. If you see a scatter plot that looks like this, the measured correlation is not telling you anything very useful. It's an artifact, even though it's mathematically correct. Right, so here what we're doing is looking at repeat acquisitions of two pictures. Here is the measured correlation. One is the Pearson, one is the Spearman. The important thing is that they're consistent. We've acquired one pair of pictures, a second pair, and so on, and the measurements change very little. However, when you look more closely, you can see that these two images, even though I've colored them differently, are actually the same. So each image is really from the same fluorophore. So if I take two images that should be identical, the measured correlation should be certainly not 0.2 or 0.3. A picture should correlate with a second image of itself perfectly. So what we're saying here is that two pictures that have been acquired that should correlate perfectly when measured aren't even close. This is about as wildly misleading as you can get. So what is the problem? What's gone wrong and what do we need to correct? Well, our problem, these are confocal images, is Poisson noise. The noise that's generated by random events. So here, typically, if we had a mean of 128 photons, this is the kind of variability you would expect in successive images for the same pixel. Our images are noisy, therefore, two noisy images, when correlated or when the correlation is measured, do not correlate perfectly, even though they should. This is a serious hole in every correlation measurement. Just to make the point clearer, I can take two images like this, which will correlate badly. If I spend more time acquiring the picture, or at least aim to capture many more photons, the images will look much more like this and will correlate perfectly, or at least more perfectly. So now we have the MAD arrangement where the quality of our correlation measurement depends on the noise level of the pictures. We have to factor this out to get measurements that are independent. Here's looking at it another way. If instead of we capture a sequence of images and we add them together so that as we're moving along here, we have more and more photons in play. And whatever these pair of images are, you can see the measured correlation rises and is going to plateau. The problem is when we've only captured one pair of images, we have no idea where in this relationship we are. We know we have a number with reassuring decimal places, but what is actually the right answer? Could we maybe take two or three images at increasing image acquisition times and try to extrapolate the plateau? Probably not very successfully. So this is a solution that we published quite a few years ago now. The argument here is that what we're doing here is we have one red image, one nominal green image, we measure the correlation. What we need is a way of measuring the noise in each picture. So the idea here is that we take two green pictures, one and then almost instantly a second one. If we measure the noise level in these pictures and do the same for the two red pictures, we can correct the measured correlation between the red and the green images. So we're using the fact that a priori we know that these two pictures should correlate perfectly to generate a correction factor. So that we take two pairs of pictures, we measure the as our noise measurement, we actually use the correlation. We measure the green 1, green 2 correlation, the red 1, red 2, combine these into a combined quality correction, which corrects the red-green measurements. And here are our corrected measurements. So even with images where the red-green measurement is all over the place, the corrected value is much more stable. I mean, there's a bit of noise down here because the image quality is very poor, but we have a mechanism for taking down here 2 pairs of poor images and producing an answer that is close to where we hope that this would eventually plateau. A very photon efficient way of making correct correlation measurements. Here we've done the same thing, but with always using the same picture, where a priori we know that the correlation should be 1, and we can see that as we increase the quality, the number of images that are joined or the acquisition time is increased, it looks as if it's going to plateau where we expect at 1. These are just showing the individual pictures. We can see here a single image is amazingly noisy. An average of 128 is looking much more respectable. Let's now throw a third problem with correlation at you. Here we have an image of a Lordenstein cell, and here we have the same image where we've massively messed around with the display contrast and offset. And when we do that, we can find these little membrane blebs. These will be bits of the membrane that have detached from the cytoskeleton and under a slightly higher internal pressure pop out like little bubbles. These are membranes under tension, so they're very thin and stretched. Here we have probably bits of plasma membrane that have much higher intensities. In this image, we can't even see the blebs, but we can see something that gives us a clue about the plasma membrane intensity. So the question then is, why is the plasma membrane here very intense, but here is much less intense? So in a cartoon form, where we have a membrane, and we're just saying, if we could blow up the pixel to see exactly what's there, we could find that a pixel is intense because there is in fact more fluorophore there, or it could be intense because there is more membrane there. So it has an average amount of fluorescence, but because there is more membrane, this pixel is more intense than that one. How is this going to be affecting our correlation measurements? This is fairly realistic because lipid bilayers are around 5 nanometers thick. Pixels in a confocal or most wide field microscopes are much bigger than that. So our problem is that the amount of membrane in different pixels varies depending on whether they're under tension or whether there is folding. So if we consider 2 fluorophores that are randomly distributed, in this picture, there will be more of both, even though they're both randomly distributed. In this one, if they were both randomly distributed, there will be less of both. So we create a correlation simply because individual pixels have more or less membrane present. Here is some simulated cells where we've made the measurements. So here is the ground truth. Here is the actual measured value. So what should be a nice negative correlation becomes 0 or positive because of the correlation created by the varying amounts of membrane in each pixel. The issue is that our region of interest doesn't fill a pixel, and it varies, and this creates a correlation of its own. So instead of reporting a nice negative correlation, we get a much more positive correlation, which is obviously somewhat misleading. The question then is how we might factor out this problem. Well, what we then need, a way of doing it, would be to have a fluorophore that reports the amount of membrane. So now we would need a 3 fluorophore arrangement. The 2 fluorophores we're interested in and one that simply reports the amount of membrane that can be then used to normalize the intensities in each pixel and get us back to the measurement we know is a ground truth. 3 problems in correlation measurement. Here's one of our many fake simulated cells that we've been playing with. If we look at the range of intensities, the amount of membrane present, we can see there's quite a big range. One way of minimizing this, at least a bit, is to take this, threshold it, reduce it to a skeleton so that we have the midline of the membrane and use those pixels. This is better, but it isn't a perfect solution. This is simply a ridiculously complex way of simulating these cells. So another way of looking at this is topography, the fact that our membranes are not flat, that they are folded. So here's the problem. The intensity in pixels originates from different volumes of membrane, different amounts of membrane in each pixel. This is exactly what we get on the edge of a solid structure. So we have a nice uniform, if we had a nice uniformly stained nucleus, the pixels at the edge would have a little bit of nucleus and a little bit of something else, which in itself would create the correlation. So the argument then is we should avoid these edge pixels, which we can in the case of a nucleus, but in the membrane, everything is an edge pixel. At no point does the membrane completely fill a pixel. Our solution then is the third fluorophore. So a kind of summary here, colocalization, we're dividing it into 2 measurements. Co-occurrence, are they in the same place? Correlation, if they're in the same place, remember we suggested that we have to only use pixels where both fluorophores are present to measure a correct correlation. The image quality is then quite important. And we've said that replicate images, 2 copies of each fluorophore, can be used to correct for noise. And here are a couple of references. Here's repeating the main bit. Colocalization, 2 separate considerations, the same place or a relationship. And that's the end of the presentation on colocalization. What we can now do is have a couple of minutes break, and then we'll look at how we might make some measurements from images that Sylvie sent me a week or so ago. But first, any questions?
Thank you, Jeremy.
No questions? It's all
I
perfectly
will
good.
wait. We'll have to be slow.
Yes.
It does happen. We have at least one person who's looking at colocalization, right? Was that you, Faye?
Hi, I'm still thinking about it because I want to use the high throughput imaging system, but it's wide field, so I'm not sure if it can be used to do the colocalization. I'm afraid not, so I would maybe regenerate my scientific questions or use other ways to do it to fix the cells on the confocal and then do the colocalization.
I mean, the issue with wide field imaging is that the out of focus fluorescence is never rejected. But an alternative, or at least an advantage of wide field, is that your image acquisition is quick compared with confocals, at least compared with the point scanning confocals that we have, rather than the spinning disks that Sylvie has. What you could do, though, is use wide field plus image deconvolution.
Which we just spoke about today.
Yeah, so try both. I mean, if you're going to do something many times, like devote months and months of work to this, compare the 2 beforehand. Find out which one is easiest and which one is also more accurate.
But I'm still not sure if it's possible to get the Z-stack using the high throughput system. I mean, if the if
You
the
mean-
vessels I'm looking for are on the different stage, maybe if I can only capture one plane, then it's impossible to explain the colocalization question.
I think for both purposes, you're better off having a Z series because you've got your cells, typically at least, many people we see come with cells sitting on cover slips. If you're taking a confocal picture and you're taking one of them, where do you take it? I mean, cells are three-dimensional structures. Depending on where the imaging plane is, you're looking at quite a different cell, really, or at least. But doing things in 3D is much harder. But if you want to do image deconvolution, you really want a 3D data set. It's also worth noting that you can deconvolve confocal pictures and appreciably improve their usable resolution and reduce the noise level. But try things and use this idea that if you take two images of the same fluorophore, they should correlate perfectly as a test to see if your measurements are correct. Try things is the real answer here.
Okay, I'll try it on the system as well, because it can capture the the maximum magnification is about 60, yeah. So I think the resolution might be good enough, and I can do the denovelation as today mentioned. But the thing is, I'm worried about if the vessels are at different levels, then I cannot explain they are colocalized with only one plane.
Again, for colocalization, are you interested in co-occurrence, that they're in the same place, or that there is an intensity relationship? I mean, co-occurrence is much easier to measure. There are far fewer problems, partly because if you're looking at the average intensity or the intensity over a group of pixels, this does improve with the number of pixels. Whereas in correlation, if the pixels are noisy, doubling the number of pixels in your measurement doesn't help.
Yeah, I don't think that you're not interested in the relationship between intensity. I think it's co-occurrence that you're.
Yeah, it's a co-occurrence.
Yeah, well, that's a much simpler measurement to make. It's far less dependent on noise. But try things. The only way you'll know is to try it. And if you want some help, you can come and talk to me or send me some pictures, or we could.
Okay, thank you.
Good. So you wanted a little, I'm just not, I can't remember what we said. Are we going to the other Zoom or do we continue here? I think we said we do go to the other Zoom, right? Or do you want us to continue here? The question is, do we need to separate the students in